Pa-share screen na lang.
Wait lang, kunin ko lang yung ballpen ko. Okay.
Sige po. Good afternoon po. Ay, parang ano na din po ito, ma'am. Parang ipe-present na din po namin. Okay
Sige.
po, sige po.
Parang rehearsal.
Okay po. So, good afternoon po. We are here to present our systematic review, the results of our extraction methods of phytochemicals in Symbopogon citratus inhibiting escape pathogens. So for introductions, here are the basic terms we need first to settle. So phytochemicals on the second slide. Phytochemicals are non-nutritive, natural occurring chemical compounds in plants which may help to improve human health. So here we have discussed further in the results terpenoid, phenolic acid, alkaloid, flavonoids, and lipids as the major categories of phytochemicals found in Citratus. Also, Citratus is a very good source of phytochemicals that is widely used and utilized throughout the tropical countries. And another important is the escape pathogens, which are the chosen microorganisms that are known for its antimicrobial resistance, and Cymbopogon citratus is looked into as one of the very good source for potential study to inhibit escape pathogens. So the extraction methods that are looked in these studies revolved in these 6 extraction methods, which are subdivided into conventional and modern extraction methods. Maceration, hydrodistillation, and steam distillation are conventional extraction methods, while vacuum distillation, ultrasound assisted, and microwave assisted are those which are improved so that there will be more efficiency in terms of extraction and biological activity. And here's an example presented in our related literature called maceration and ultrasound assisted. And the method is called maceration. The extracts found of phytochemicals are flavonoids, carbohydrates, tannin, alkaloids, steroids, and phytosteroids. And this acts against SRUs, and the inhibition is at intermediate 50 milligram per ml of zone of inhibition, and same as the ultrasound assisted extraction method. So for the significance of this study, our aim is to evaluate the suitability of various extraction methods for the phytochemicals of Citratus inhibiting escape pathogens and provide significant information that will benefit to public health policymakers, agencies for drug development and pharmaceutical industries, and the academe for scientists and future researchers. So the major general objective of this study is to have a collective discussion on the different extraction methods and phytochemicals in Citratus that inhibits escape pathogens. And in order to find that, we have these goals. Number 1, to gather and compare conventional and modern extraction methods for Citratus in terms of extraction yield, phytochemicals extracted, and effectiveness against escape pathogens. And number 2, to identify the phytochemicals that are found bioactive against escape pathogens. And third, to determine the extraction methods of Citratus phytochemicals that inhibit escape pathogens. So to run through this study, we have used the Prisma protocol, and here is the processes that are involved. So to trim it down, first we have identified the databases and the registers. In the figure 6a, it is found the major databases of Google Scholar, Science Direct, and Semantic Scholar. However, we have also utilized other sources, and the number of results from databases are presented in the table, including the finalized keywords that are used. And on the figure 6b, here is the process of screening. There is the inclusion and exclusion, but prior to that, duplicates were removed, and also um, those which are not experimental and not in full text access will also excluded. And then the further details of inclusion and exclusion method is presented in figure number 7. So here, a series of questions were asked, and if any of the studies were not do not pass all these questions, they are not included. But for those, they are eligible for the further steps of this study. So for those which are eligible, they undergone the risk of bias. So for the risk of bias, there are many tools that are available for systematic review. However, the OHAT tool is the one that suits this study because it identifies with the in vitro type of study specifically in our research. So this is the table that presents the eligible data studies which were assessed for the risk of bias. So out of 25 studies which were eligible, only 22 acquired a definitely low rating and passed the assessment. And these 22 studies were further uh, assessed for its quality appraisal using the Saimago tool. And out of 22 low risk of bias, only 15 got accepted because the quality of publishers of some of the papers were not found in the Saimago website, and also only up to quartile 3 were accepted in this study. And here is the final results of the accepted screen data studies. So there were 15 studies, and the profiles were also presented in table 5. 15 data studies were to be further analyzed after extraction of the data and will be processed, presented, and be analyzed. For the results, may I call MJ?
So for our first presentation of results, it's the comparison between the extraction methods of conventional and modern. Out of 6 extraction methods, we found 3 for the conventional, which are maceration, hydrodistillation, and steam distillation. And for modern, we have vacuum distillation, microwave-assisted extraction, and ultrasound-assisted extraction. Maceration was utilized by 3 studies presented here at the left side, which are from from Cherian and Cherian also, because the solvents used were considered. And for hydrodistillation, 7 studies also are presented from Nasim 2,
Nazim, Tuwaza, Desai and Parik, Sonobu, Fadli, Owolabi, Simanga, and Simanga. And for this team's distillation, Chalchat, Salim, and Bugayong and their group. And now for the modern methods. Two studies have resulted with this extraction method from Falca and Viktorova. Two also from MAE, which are from Blese and Parik and Tadong and their group. And UAE is resulted from Nasim's paper. So we have observed that ethanol extract and hexane extract from conventional method, specifically maceration, resulted with the highest yield of 9% and 6.60%, while vacuum distillation was the highest for the modern extraction methods, which is specifically 3.40% for Falcaco. Next. Next is the extraction methods and phytochemicals that are compared and presented in our Venn diagram. These answers are objective number 1, letter B, which are the phytochemicals present in the extracted phytochemicals in the methods. So the common extracted phytochemicals are the terpenoids, specifically nerals, citral, Z-citral, geraniol, E-citral, beta-myrcene, and for the phenolic acids, we have flavonoids. Interestingly, we also observe that there are unique phytochemicals from hydrodistillation, which are the lipids, neryl acetate, and phenolic acids, eugenol. Also for ultrasound-assisted extraction as a modern method, we also have phenolic acids and flavonoids po. Next, ikaw na po, Richard.
So, for the extraction methods and inhibited escape pathogens, for the conventional extraction methods, most conventional methods inhibited So for the extraction methods and inhibition of escape pathogens, inhibition of S. aureus was mostly effective using the modern extraction methods, specifically the ultrasound-assisted extraction, as shown in Figure 11. While on the other hand, conventional methods resulted in the resistance or less inhibitory activity against S. aureus. Likewise, inhibition of the Pseudomonas aeruginosa was higher in modern extraction methods than in conventional extraction methods. The pathogen, which is the Pseudomonas aeruginosa was mostly resistant to hydrodistillation and steam distillation. While for Klebsiella pneumoniae, there was an intermediate activity observed in the Sisystratus phytochemicals using maceration with the ethanolic extract, and a susceptible activity employing hydrodistillation. However, um, there is a resistant activity in the in some studies which tested pneumoniae. On the other hand, extracting phytochemicals from Sisystratus through hydrodistillation was effective against the Acinetobacter baumannii as it resulted in susceptible activity. So phytochemicals that are derived from plants are considered to be bioactive compounds. So in this systematic review, we have we've selected studies that focus on phytochemicals from Sisystratus using different kinds of extraction methods against four escape pathogens which are S. aureus, P. aeruginosa, pneumoniae, and Acinetobacter baumannii. In conventional methods, S. aureus, P. aeruginosa, pneumoniae, and A. baumannii were tested, while only S. aureus and P. aeruginosa were tested for modern methods. This table presents the various extraction methods utilized in the collected studies. For the conventional extraction methods, compounds extracted from hydrodistillation, which are terpenoids and trace amounts of phenolic acids, were bioactive against S. aureus, P. aeruginosa, and Acinetobacter baumannii, as well as Klebsiella pneumoniae. There were also, in maceration method, there are also case studies that utilizes the maceration method also obtained different bioactive compounds including terpenoids, lipids, phenols, tannins, saponins, alkaloids, and flavonoids that have shown bioactivity against S. aureus, P. aeruginosa, and K. pneumoniae. On the other hand, in the modern methods, major phytochemicals are found were terpenoids and phenolic acids, specifically the flavonoids. In the two studies that used the vacuum distillation, terpenoids were observed to have bioactivity against S. aureus and P. aeruginosa. Lastly, the ultrasound-assisted extraction showed the presence of phenolic acid and flavonoids with bioactivity against S. aureus. Based from the result, the terpenoids, which is the highest phytochemicals extracted from most of the extraction methods, were bioactive against S. aureus, P. aeruginosa, P. pneumoniae, and Acinetobacter baumannii. Diyan po sa last.
Dito naman po, for modern extraction methods, also presented that majority of the extraction methods that are used have almost similar na extract na bioactive compounds, namely the terpenoids. But for ultrasound assisted extraction, it has flavonoids, and as we can also observe for some studies, their compound yield percentage were not stated, so we have just put the not stated statement there. And also, if it is also observed in this table that majority of the modern extraction methods has inhibited the S. aureus and P. aeruginosa escape pathogens. For the next table, the next table presents the inhibition against escape pathogens. So here we have uh, found whether the extraction method and the escape pathogen were inhibited or not through the use of zone of inhibition or minimum inhibitory concentration. So we have separated the tables for the zone of inhibition and minimum inhibitory concentration, but for this slide, it these were the ones that were tested for zone of inhibition. They are maceration, hydrodistillation, and steam distillation for the conventional extraction methods. There were three that does not inhibited the pathogens of Klebsiella pneumoniae, Pseudomonas aeruginosa, in the extraction methods of maceration using hexane solvent and also maceration using the ethane solvent, and also the maceration using as well the hexane. And for the hydrodistillation, only one that in the name of P. aeruginosa did not inhibited, and it was also resistant of its uh, control used, which was gentamicin. While for the steam distillation, there were two that does not inhibited, which were S. aureus and also P. aeruginosa, together with their low zone of inhibition. For the next slide discusses the minimum inhibitory concentration. So for hydrodistillation and steam distillation, they are only the ones that use the MIC, but there is no data found in the review presenting as well of the maceration. So for hydrodistillation using MIC, majority were does not show inhibition, that does not inhibited the escape pathogens being tested for. However, it is also found here that hydrodistillation has inhibited the Acinetobacter baumannii, which were susceptible in their epiminem control. While on the steam distillation, all of the uh, escape pathogens does not inhibited using the MIC. For the table 10, so here the modern extraction methods were found to that inhibited to all the extraction methods presented. However, only S. aureus and P. aeruginosa were found to be the focus of these extraction methods. But their results are good because they have inhibited the pathogens. So for the conclusion and recommendation, generally, conventional methods extracted more Cicitratus yields and phytochemicals. On the other hand, modern methods inhibited the escape pathogens more effectively than the conventional. And major bioactive compounds inhibiting escape pathogens found in Cicitratus were terpenoids, flavonoids, phenolic acids, tannins, saponin, and alkaloids. For the recommendation, it is recommended that future similar studies must have a longer time frame to allot for each systematic review processes, and the inhibition test of Cicitratus against all escape pathogens, particularly on Enterobacter species and Enterococcus faecium. And then lastly, complete data should be present in data studies, specifically extraction yield, quantified phytochemicals, and controls for assessing MIC or the zone of inhibition. So that's all. Thank you.